Welcome to HIV and the immune system. So let's crack on. So here's a little diagram. It asked in the exam what A and B were. So A is our lipid envelope. Label this up. It's not a cell membrane. It used to be a cell membrane, but it is no longer a cell membrane. This isn't a cell. HIV is a virus, and it is acellular. It's not a cell. It doesn't transfer energy. It is not a cell. So A is our lipid envelope. Embedded in the lipid envelope are our antigens, but to the virus, these are attachment proteins. It's how they attach to a certain cell. Yeah, these are antigens, as in if they will trigger antibody generation if they get into a cell. B is our capsid. So that's a protein. If you ever get a proteins essay, you can talk about the capsid. No, I'm not sure how much you can talk about it. This dot here, and it's the same below, that's an enzyme. And this is, you need to know the name of the enzyme. This is reverse transcriptase. Enzinase, it's an enzyme, it does the reverse of transcription. And this is its genome, and its genome is RNA. So this is RNA, and that's its genome. Contains its genes. It's repeated underneath. This space here hasn't got a name. It's just it'll just be fluid. This is also a protein. It's not on the syllabus though, but if you're keen to have some off-spec knowledge for a future essay, this is the tegument. Again, there's not many words that begin with TEG, apart from teg. But anyway, yeah. So that's tegument, and that's also another protein. So if it was like a proteins importance of proteins essay, you've got three proteins to go at here. You've got the capsid protein, the tegument protein off spec, and reverse transcriptase is an enzyme. It's therefore a protein. So it might make a nice paragraph in an essay. So this is HIV, and it crucially its attachment proteins means it can bind to and get into T helper cells. So yeah, so this infects helper T lymphocytes or T helper cells, helper T cells. You can say it either way around. So let's assume it's got into a T helper cell, and it inserts its RNA, and then it just uses its enzyme, reverse transcriptase, to form DNA. It does the reverse of transcription. Transcription is making mRNA from DNA, so we just do the opposite. Yeah. DNA will then integrates into the nucleus. So this, integrates into TH cells DNA chromosomes. It uses an enzyme called integrase, off spec. It's also in the virus, but so that means that the cell is permanently infected with HIV. Yeah, so it's got the genome of HIV, and T helper cells, these TH cells live for a long time, decades. And then we're going to make mRNA from it. So now, if we've done reverse transcription to get from RNA to DNA, this is just transcription. Again, I'm not going to go through it now, but hopefully you can write about, RNA polymerase joining together RNA nucleotides by phosphodester bonds. And then we do translation. Yeah, this is the cell's ribosomes. Squeeze it in. And we make the structural proteins like I put above, the tegument, reverse transcriptase, the attachment proteins, the capsid proteins, all those are like structural proteins, and the enzymes are also made. So reverse transcriptase is made, also DNA integrase is made, again, not on the syllabus. And then they all kind of come together, and then the HIV bud off from the cell. Okay, and as they do, they get coated in the lipid envelope. So if we just put here, they get coated in the lipid envelope. And remember, lipids are fluid. Well, phospholipids are fluid. They aren't attached, and therefore, because the HIV has a phospholipid bilayer and the cell membrane is a phospholipid bilayer, when you push them together, they will fuse together, and that's how HIV infects the cells. It binds to T helper cells because of its antigens, and it will then fuse with the cell because of the lipid envelope. So let's assume someone's got infected with HIV. So normally, the classic was a lot of intravenous drug users got HIV in the 80s because they were sharing needles. So someone's got HIV, they share a needle, and they inject HIV into themselves. And I think it's important to remember that it's not just one HIV going in. There will be millions of HIV going into a person's blood if they injected it in, or like unprotected sex as well that causes injury, and therefore blood transfer. It has to be blood transfer. So yeah, so we've got HIV going into the body. Now this is going to have two kind of like two things can happen here. One, it can infect TH cells. Yeah, that's what it wants to do. Want as in it hasn't got a brain, but that's if it bumps into a T helper cell, it'll infect it. So some of them are going to infect TH cells, but the rest is just like ambling around the body, hoping to bump into a cell. Well, they'll probably end up being phagocytosed. Phagocytosis might take place. So a phagocyte will bump into it, see you're foreign, and they're going to attack. And then we can go through this entire immune response that phagocytosis can take place. Then we get antigen presenting cells. Antigen presenting cells are made, APCs. Then we do something called clonal selection, where we look for a B cell and a T cell with a complementary receptor. Yep, so that's when we then activate B and T cells, that have a complementary receptor to the antigen. Now this is where more TH cells are needed. TH cells secrete cytokines, which is a chemical stimulant. It stimulates the cells to activate. These B cells and T cells are dormant, and they're just waiting to be activated. So then we undergo clonal expansion. Let's stick in blue. One here. So now we do clonal expansion. And you talk about mitosis here as well. So we get an army so we're going to get plasma cells from the B cells. We're also going to get cytotoxic T cells. Oops. Cytotoxic T cells, and we're also going to get memory B and T cells. Okay, and plasma cells, these are going to secrete antibodies. Cytotoxic T cells, these kill infected cells. Which is bad news for our T helper cells. And then the memory B and T cells, these are going to provide immunity. However, it don't work for HIV. Yeah, normally that would work. So yeah, normally we'd shift the infection. So then, and I would focus on the antibodies just in terms of, because we can kind of give more detail of how these work. So these are going to then agglutinate HIV in blood, any that are roaming around their body, and then they're going to be phagocytosed. So it's just easy phagocytosis is going to take place. So this should clear the infection. The plasma cells make the antibodies. Antibodies are going to mop up any HIV in the blood, and the cytotoxic T cells are going to kill any infected cells. However, my infected cells are my T helper cells. So if I kill my T helper cells, I can't do this. Yeah. So some T helper cells will be destroyed, but others are going to be, in the process of just, the virus is in there. There's no evidence of the virus in there yet, and the cells live for a long time. They're going to replicate inside the T helper cell, and burst out, and therefore this process is going to repeatedly happen over time. Okay, and every time it happens, I'm just depleting my T helper cell count. Yeah, so I need a critical number of T helper cells to have an effective immune system. So as HIV bursts out, it's going to be destroyed, but some are going to get into fresh T helper cells and then replicate inside them and burst out, and so on and so on. It can take 10 years for the immune system to collapse enough that you get symptoms of the virus, which is AIDS. I mean, the first symptom of the virus is a fever. So you would, inject the HIV from contaminated blood, and it would cause a fever. Which kind of shows that the immune response is happening correctly. But then it's just this slow depletion of T helper cells. So let's just finish by putting that. I'm doing okay for time for one, so let's put a little asterisk up here on T helper cells. So this is over many years. The number of TH cells decreases. Therefore, can't complete clonal selection for any pathogen. Therefore, can't fight infections, fight pathogens. Therefore, many infections, which equals AIDS, which is acquired immune deficiency syndrome. So the immune system is becoming deficient because we are losing the T helper cells. So I think a lot of the misconceptions come from the idea that people think that like one HIV comes in. Yeah, it doesn't. There's going to be many. Some are going to get into T helper cells. Some undergo phagocytosis. You do the immune response, make antibodies, agglutinate HIV, stick them together. In other words, and then stimulate phagocytosis. So we're really effective at removing them from the blood. However, HIV is hunkering down in these long-lived T helper cells, slowly replicating, bursting out occasionally, reinfecting T helper cells. So the T helper cell count decreases and decreases and decreases. And when it gets to a critical level, you can't complete this process here, clonal selection, and therefore you can't activate the B and T cells. One last point on T cells. They're called a T cell because they mature in the thymus gland. So a T helper cell and a normal T cell, that's all they have in common. They have completely different jobs. So don't confuse a T cell with a T helper cell. Okay, so that's it done. Under 15 minutes for once. Well done me. I shall have a coffee to celebrate. Take care, everybody.